This is a chapter of biomolecules. It's a very interesting chapter. Like just carbon, hydrogen and oxygen. These are the main things. They just unite together with a little bit of sunlight. It is converted into literally non-living organism, but they can do living activities. Some will have some metals and other things, right? So biomolecules, it's an amazing, wonderful thing that we need to understand. But it's not an easy chapter, so I'm going to take you through very slowly. Like let us see how we analyze the organic compounds. Just how we chemically analyze organic compounds. Organic compound is like just a flush. To analyze the chemical composition of the organic compound of a living organism, the chemical analysis of the living tissue is done. As organic compounds play a major role, it is essential to have knowledge of their molecular formula and structure. It is essential to have the molecular formula and structure. This can be done by performing the steps which I am going to tell you. First is the weight of the living tissue. Like a piece of liver, fruit or vegetable of any other body tissue is taken. Just the weight. Next is the tissue is then grinded in the solvent called as trichloroacetic acid. Trichloroacetic acid with the help of the mortar. So with the help of the mortar, which we can just grind it. This thick slurry is obtained, is then filtered through a cheese cotton or just a cotton. Just you have the cotton cloth, you can just do it. This will generate two fractions of solution. There will be two fractions of solution. The filtrate or the acidic soluble fraction, retentate or the acid insoluble fraction. There are two different things. One is the filtrate and another is the filtered. The extracts can then be subjected to numerous separation techniques. It can be subject to numerous separation techniques in order to obtain the desirable compound from all other components. We can get an idea of the molecular formula and the portable structure of the compound. It is that simple. Okay. So liver can be analyzed, eyes can be analyzed, any tissue can be analyzed in fact. Okay. So next is all the carbon compounds that we can get from the living tissue are called carbon molecules. Next is the ash analysis of the inorganic compounds. Inorganic compounds. And the elements in the ash is the remaining part of all living tissue which remains after burning of any organic compound. If you burn any organic compound, you will find the ash that you are analyzing it. Ash analysis is done in order to analyze the chemical composition of different inorganic compounds and elements present in the organic materials after burning. So after burning it, you just get the organic materials. It is done by one, for analysis, a small amount of living tissue is taken, which is oven dried. You put it in the oven and it is dried till all the water evaporates. This gives dry weight. You know the weight of it, so it gives a dry weight. After that tissue is burnt completely, which results in the formation of ash, ASH, ash. All the carbon compounds are oxidized to gaseous form, that is carbon dioxide, water vapor, and are removed. Thus, ash formed contains inorganic elements like potassium, sodium, iron, calcium, magnesium, you name it, all those kind of things, right? So we have taken out the carbon dioxide, we have taken out the water vapor in it, okay? The inorganic compound are also seen in the acid soluble fraction. It is also seen in the acid soluble fraction. Cellular pool, the sum total of different types of biomolecule compound and ions present in the cell is called as cellular pool. It contains more than 5,000 chemicals. It contains more than 5,000 chemicals. Biomolecules are small in size with low molecular weight, 18 to 1800 Dalton, highly soluble, and have a simple molecular conformation. Micromolecules can be inorganic, such as water, mineral salts, gaseous or organic compounds such as sugar, amino acids, nucleotides, etc. All these compounds mentioned are soluble fractions of filtrate except the lipid, which occurs as an insoluble fraction of the filtrate, as they are mostly found in the cell. They are mostly found in the cell membrane and thereby form vesicles. Thereby forms vesicles which are separated as an insoluble pool, Carbohydrates will be there. These are the organic compounds mainly made up of carbon, hydrogen and oxygen. They are defined as polyhydroxyaldehydes and ketones. Carbohydrates are also known as saccharides because their major constituents are sugar. These are divided into following two types. Monosaccharides, that is number 1. They are simplest carbohydrates which cannot be hydrolyzed, further into smaller components. These are generally composed of 3 to 7 carbon atoms per molecule. 3 to 7 carbon atoms per molecule. Monosaccharides are also known as reducing sugars because they have a free aldehyde or the ketonic group and can also reduce to Cu, that is cupric ion. Copper, that is what we call as cupric ion of the Benedict's or failing solution to cuprous ion. For this we can give an example is ribose, glucose, all these kind of things. Next, oligosaccharides. We have seen the monosaccharides, now it's the oligosaccharides. Oligo is minimal, that's what is oligo is. These are formed by condensation of 2 to 6 monosaccharide molecules. The bond between two monosaccharide units is called a glycosidic bond. Disaccharides, these are sugars containing two monomeric units and can be further hydrolyzed into smaller components. These are known as non-reducing sugars. Because the free aldehyde or ketone group is absent. Example, sucrose, maltose, lactose, etc. Okay. Next we will move on to trisaccharides. These contain 3 monomers. Example is raffinose. Next is tetrasaccharides. Tetrasaccharides is the saccharose. You don't have to remember that name, but I'm just giving you tetrasaccharides and so on. Okay. So we have finished the carbohydrates. Amino acids. Amino acids are the organic compounds containing as amino group and acidic group as substituents on the same carbon. The carbon which occupies the 4 different valency position. These are hydrogen, carboxyl, amino and R group. The general formula is shown in the picture. Okay. So where the R is equal to side chain of the amino acid. So R is equal to the side chain of the amino acid. The amino group accepts a proton, whereas the carboxyl group donates a proton. Proton is a positive ion. Donates a proton. So, an amino acid can act as both acidic and base. Acidic and base. Hence, it is a amphoteric in nature. The chemical and physical properties of amino acids are essentially due to amino carboxyl and R functional group present. Okay. Acidic amino acids. These contain one amino group and two carboxyl group. One amino group and two carboxyl group per molecule. Example, glutamic acid and aspartic acid. Next is basic amino acids. These contains two amino group and one carboxyl group per molecule. Example, arginine, lysine and histidine. Neutral amino acids. These contains one amino group and one carboxyl group per molecule. Example, methionine, isoleucine, serine, threonine, cysteine, glycine, alanine, valine, leucine, asparagine, glutamine and proline. You need to learn all these amino acids. Probably over a period of time, you will automatically know about it. Next is the aromatic amino acids. These contain aromatic ring, their side chain. For example, for these things are phenylalanine, tyrosine and tryptophan. Okay, that finishes amino acids. Now we are moving on to lipids. Lipids are esters of fatty acids and alcohol. These are generally insoluble in water. They could be simple fatty acid. Simple fatty acid or organic acids having hydrocarbon chains that end in carboxylic group. Palmitic acid has 16 carbons including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl carbons. Depending upon the types of bonds present, fatty acids are of the two types. One is saturated fatty acid. Fatty acid which do not have double bond. Okay. Double bond is C. These are generally solid at room temperature. Unsaturated fatty acids. Fatty acids which contains one or more than one double compounds. Next is the simple lipids. These are esters of fatty acids and various alcohol. There are two types. A, neutral or the true fat. These are esters of fatty acid with glycerin. They are also called as glycerides. Next is waxes. You have in the ears, isn't it? That is what waxes. These are esters of fatty acids with alcohol other than glycerol. Next is compound or conjugated lipids. These are esters of fatty acids and alcohol but contains other substances also. Okay. Example, phospholipids, glycolipids, cutin, suberin, etc. Next is phospholipids or lipids which have phosphorus and phosphorylated organic compound in them. One of the common example of phospholipid is lecithin. This is a common example of phospholipid is lecithin. Derived lipids. Now we want to learn about the derived lipids. These are lipids like substance such as sterol and derivatives of lipids. Example is steroids, prostaglandins, and these are the two main things which we need to know, steroids and prostaglandins. Fats are also differentiated into two main types on the basis of their melting point at room temperature. Melting point at room temperature. Number one is the hard fats are solid at room temperature. Hard fat are solid at room temperature and contains long chains of fatty acids. Long chains of fatty acid. Example, animal fat. Okay. Oils are usually liquid at room temperature because they have low melting point. Example is groundnut oil, cottonseed oil, mustard oil, almost all the oils. Okay. Oil is non-polar and is not attracted to water. They are not attracted to water. So oil does not dissolve in water. Oil do not dissolve in the water. Moving on to the next slide. These are the monomers of nucleic acid. Monomers of nucleic acid. The nucleotides are made up of 3 molecules. The nucleotide is made up of 3 molecules. Pentose sugar, a cyclic nitrogenous base, and a phosphoric acid. Cyclic nitrogenous base and a phosphoric acid. That is the phosphate group. Okay. Next is adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidalic acid. Okay. Moving on is the pentose sugar. It occurs in pentagon or Frobenius form with 4 carbon on 1 oxygen forming a ring. It is present in the form of ribose deoxyribose sugar in RNA and DNA respectively. Okay. Nitrogenous bases. These are flat heterocyclic compounds having nitrogens and carbon in a ring structure. These are basically of 2 types. One is purines, Purines. It is larger and composed of 2 rings. They are further of 2 types. Adenine and guanine. Next is pyrimidines. It is smaller and composed of single ring. They are of further 3 types. Cytosine, thymine and uracil. Phosphoric acid, phosphate group, phosphoric acid. It is composed of phosphoric acid. A nucleotide may have 1, 2 or 3 phosphate group. A nucleotide may have 1, 2 or 3 phosphate group. It gives acidic nature to the nucleotide. Next slide. A larger number of organic biomolecules are present in the cells which are used in various metabolic reactions of the cell. These are divided into 2 types. One, primary metabolite. These are metabolites which are found in animal tissues. These functions are easily identifiable. They play specific known roles in the normal physiological process. For example, amino acids, carbohydrates, proteins, nitrogen bases, all these things they play a normal physiological process. Next is the secondary metabolites. These are metabolites which are generally found in plants, fungal and microbial cells as a byproduct of main metabolic reactions. They are the byproduct of the main metabolic reactions. Their functions are not identifiable in host organism and are not yet understood. Example, alkaloids. Alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigments, scents, gums, spices, all these things. Okay. So what are their functions? Both primary and secondary metabolites serve many functions. Let us see few of them. Many of them are used for welfare of the human being. Example, rubber. Some of the drugs, many species, scents, pigments. Some have ecological importance. Moving on to biomacromolecules are large in size, higher molecular weight, and the molecules about 10,000 Daltons and above. 10,000 Dalton and above. These are generally formed by linking a number of micromolecules, commonly known as monomers. All these compounds are found in the acid insoluble pool. These are of 4 major types. Example, protein, polysaccharides, nucleic acid and lipids. The molecules which are found in living organisms are divided into 2 main types. One is biomicromolecules. These are molecules which have their molecular weight. Molecular weight less than 10,000 Daltons. Next is biomacromolecules. These are molecules which have their molecular weight 10,000 Dalton and above. All these macromolecules are actually polymers of their biomicromolecules. Example is polysaccharides or polymers of the monosaccharides. They are the polymers of the monosaccharides. Proteins are polymers of amino acids. Nucleic acid are polymers of nucleotides. Homopolymers are those which have only one type of monomers. There are only one type of monomers present. These monomers can be repeated in number of times in a chain. They can be repeated number of times. For example, insulin. The next example, starch. Remember the examples. Those are the things that be asked in the multiple choice question. Okay. Heteropolymers. Heteropolymers are those which have 2 or more than 2 types of monomers. 2 or more than 2 types of monomers. Example, protein. Various macromolecules and their major role are described. Next is lipids. The molecules in the insoluble fraction are polymeric substances except lipids. Although lipids have their molecular weight not exceeding above 800 Dalton. But still it comes under acid insoluble fraction. Example, biomacromolecule categories. This happens because these are small molecular weight compounds and are present not only as such but also arranged in structure like cell membranes and other membranes. Okay. The cell membranes and other membranes are broken down into pieces and form vehicles that are not water soluble. Therefore, these are separated along with the acid insoluble pool and are placed in macromolecules. Lipids are not strictly biomacromolecules. They are not strictly biomacromolecules. If representation of chemical composition of living tissue is done from abundance point of view and arranged class-wise, it is observed that water is the most abundant chemical in living organism. Water is the most abundant chemical in the living organism. I repeat, water is the most abundant chemical in the living organism. These are the most important and abundant intracellular organic biomolecules. These are polypeptides having chains of amino acids arranged linearly that are linked by peptide bonds. Linearly that are arranged by the peptide bonds. Each protein is a polymer of amino acids. Each protein is a polymer of amino acids. There are 20 types of amino acids. There are 20 types of amino acids. I'll just name few of them, but you need to read everything. Alanine, valine, arginine, leucine, histidine. Okay. Proteins are considered as a heteropolymers. Put heteropolymers and not homopolymers. Okay. These amino acids are divided into two main types. Essential amino acids. These are amino acids are essential for our health. So it's called essential amino acids. The dietary proteins are of the source of essential amino acids. Whatever that comes from the diet, it is called as essential amino acids. Example, leucine, isoleucine. Then moving on is the non-essential amino acids. Non-essential amino acids. These are those amino acids which are our body can synthesize itself. Our body itself can synthesize. Example is proline, serine, and etc. There are few others. Proteins are heteropolymers containing strings of amino acids. Okay. Biologists describe the protein structure of 4 different levels. Biologists describe it, there is a 4 different levels. One is the primary, secondary, tertiary and quaternary. So, primary, secondary, tertiary and quaternary. Primary, the primary structure, it is the description of the basic structure of the protein. Description of the basic structure of the protein. This includes number and sequence of amino acids in each polypeptide. The distance between two adjacent peptide bonds is about 0.35 nanometers. 0.35 nanometers. A protein is imagined as a line whose left end is represented by first amino acids, also called as N-terminal amino acids. N-terminal amino acids, and the right end is represented by the last amino acids called as C-terminal amino acids. It is called as C-terminal amino acids. Example, insulin, ribonuclease. Insulin, just remember insulin. Next is the secondary structures. Secondary structure. The thread of primary protein is folded in the form of helix. This is stabilized by hydrogen bond between oxygen of the carboxyl group of one amino acid residue and NH group of the next fourth amino acid residue. It is very difficult to follow, but you will remember those things when you are doing an MCQ. Okay, so just listen to it. Example, keratin. In beta-plated secondary structure, 2 or monopolypeptide chains get interconnected by hydrogen bonds. Let's look at the tertiary structures. There is a bending and folding of various types of forms, a hollow woolen ball like sphere, rods or fibers. Okay. Tertiary structure is stabilized by several types of bonds, hydrogen bonds, ionic bonds and van der Waals interactions, covalent bonds and hydrophobic bonds and hydrophobic bonds. It gives information about a three-dimensional configuration to the protein molecules. Example is myoglobin. Quaternary structure. Quaternary structure. Certain proteins consist of an assembly of more than one polypeptide or subunits. Thus, the individual polypeptide or subunits are arranged with respect to one another. They are arranged with respect to one another. Example here is hemoglobin. Hemoglobin is the best example. Other examples are lactic acid, dehydrogenase enzymes. Okay. Then moving on to proteins are classified as basis of shape, chemical composition and functions. Proteins are classified on the basis of the shape, chemical composition and functions. These are of two main types. One is fibrous protein, the protein having spiral secondary polypeptide chain wound around each other in order to form fibers. These are insoluble in water generally. But soluble in concentrated acid, alkalis and salt. Concentrated in acid, alkalis and salt. Example, collagen of connective tissue, keratin of hair, etc. Okay. Next is globular protein. They are rounded in shape. And are generally soluble in water and in dilute acid. They are soluble in water and dilute acid, alkalis, salt. Alkalis and salt. Example for this is egg albumin, serum globulin. Okay. Now, proteins have a various basic function in living organisms. They have various functions in the living organisms which are helps in transportation of nutrients across the cell membrane by acting as protein transporters. And then helps in fighting with infectious organisms. They are also helpful in movement of the muscles. Example, myosin and actin. Helps in the maintenance of the pH and regulation of volume of the body fluid. Helps at the time of injury in blood clotting. And acts as antibodies and provide immunity. Next is it helps in growth and repair of the body tissue. Some proteins function as hormones and some functions as enzymes. And catalyzes the reaction. Denaturation of proteins. When proteins are exposed to extreme change in pH, acid or temperature, the weak bonds holding the tertiary and quaternary structure get disrupted so that the protein unfolds. This unfolding is known as denaturation of protein or loss of its functioning. So, denaturation of protein or loss of its functioning. Denaturation is not strong enough to break peptide bonds. Thus, primary structure remains unaffected. Next slide. These are another class of macromolecules and are present in the acid insoluble fractions. Polysaccharides are long chemicals of sugars. We all love sugars, aren't we? They are not sweet and insoluble in water. Polysaccharide chain is made up of two ends whose right end is called reducing end and the other left end is called non-reducing end. It is called as non-reducing end. The next slide. These are two types. Homopolysaccharides. Homopolysaccharides. These are those complex carbohydrates which are formed by polymerization of only one type of monosaccharides. Monomers. One saccharides are monomers. Examples: starch, glycogen and cellulose. Starch, glycogen and cellulose. Cellulose, it is a polymeric polysaccharides which consists of only one type of monosaccharides. Only one type of monosaccharides, monomers. That is, glucose, it is known to be a rigid and insoluble polysaccharides. Insoluble polysaccharides found in cell wall of most algae, certain protists, fungi and all higher plants. As cellulose is not composed of complex helices, so it cannot hold iodine. It cannot hold iodine and cannot give color with iodine. Next is starch, a storage polysaccharide because it helps in storing energy in plant tissue. Chemically, the starch is formed of two glucose monomers. Example: amylose and amylopectin. Amylose and amylopectin. Such forms helical secondary structures. Glycogen, it is also storage polysaccharides found in animals only. It is also known as animal starch. Glycogen is also known as animal starch. Inulin, it is a polymer of fructose. It is a naturally occurring polysaccharides produced by many types of plants. Some plants store food in the form of inulin. Some plants are store food in the form of inulins. Example: sweet potato, agar, xylon, arabam, etc. or some of the types of homopolysaccharides. This is some of the examples of homopolysaccharides synthesized by algae and some plants. Next, let us look at the heteropolysaccharides. These are complex carbohydrates formed by polymerization of two or more than two types of monosaccharides. Monomers. Example: chitins, pectins, peptidoglycan, hyaluronic acid. Chitins. It is the second most abundant natural polymers found in exoskeleton of arthropods. Arthropods, joint legs, right? It's a prawn, crab, etc. And in cell wall of fungi. It has building blocks of amino sugars and chemically modified sugars. Building blocks of amino sugars and chemically modified sugar. It is made up of N-acetyl glycosamine unit interlinked by glycosidic bond. Next slide is: polysaccharides play multiple function as described. They play multiple functions. Number one, it acts as structural compounds in cell wall of plants, certain fungi and protists. Example: cellulose, chitins help in anticoagulation and prevents blood clotting inside the vessels. Example: heparin helps in lubrication of joints between bones. Lubrication joints between bones. Example, hyaluronic acid. Example, hyaluronic acid also used in tissue culture. It is used in tissue culture. Example, agars. So all the agars which we do is blood culture, those kind of things. Acts as a reserve food materials in the plants. Reserve food materials in the plant. Example is starch. Other types of macromolecules found are part of acid insoluble fractions of any living tissue is the nucleic acids. These are polymeric compounds of nucleotides. Polymeric compounds of the nucleotides. Example, polynucleotides. A nucleotide is composed of three chemically distinct components. Three chemically distinct components: heterocyclic compound, nitrogen base, adenine, guanine, uracil, cytosine, and thymine. The skeletal heterocyclic ring DNA RNA is called as purine and pyrimidines. Purine and pyrimidines. Adenine and guanine are purines, while uracil only in RNA. Uracil is present only in RNA. Thymine only in DNA. In this you got to mug up. Uracil is present only in RNA. Thymine is present only in DNA. And cytosine are pyrimidines. Monosaccharides, ribose or deoxyribose, phosphoric acid or phosphate. A nucleic acid which contains deoxyribose sugar is called as deoxyribonucleic acid. It is a DNA. Deoxyribonucleic acid, while that contains ribose sugar is ribonucleic acid, which is the RNA. So that is so simple. Okay, it's been so complicated, but if you listen to this 3 or 4 times, it will just get into your brain, and when you do the multiple choice question, it will automatically come for you. Okay, that is what is important in this multiple choice. DNA. DNA is genetic material found in the nucleus of all living cells except some viruses. So viruses do not have DNA. Structure of DNA. The structure of DNA was elucidated by Watson and Crick based on X-ray and diffraction studies. Just remember the X-ray diffraction studies. They proposed a double helix model of DNA. Double helix model of DNA. DNA exists as a double helix and consists of 2 strands of nucleotide that are anti-parallel to each other. They are anti-parallel to each other. Parallel is this, but it's like an anti-parallel to each other. Both run in opposite direction. One is 5-3 direction, the other is 3-5 direction. DNA is formed by sugar, phosphate, sugar chain. The nitrogen base are protected more or less perpendicular to the backbone of DNA and faces inside. A and G of one strand base pairs with the T and C respectively on the other strand. Okay. Between A and T, there are 2 hydrogen bond, while there are 3 hydrogen bond between G and C. Okay. So just remember that. Okay. DNA has a uniform thickness of 20 angstrom and pitch is at 34 nanometer. Thus, 1 turn of DNA measures 3-4 nanometers. This form of DNA is called as beta DNA. This form is called as bDNA. Okay. Nucleic acid. Nucleic acid play a multiple role in living organisms. These are given as 1. It enables cell to grow, maintain and divide by directing the synthesis of structural proteins. Synthesis of the structural proteins. Next, they act as a genetic material, transfer hereditary characters from one generation to the next. Characters from one generation to the next. Okay. Moving on to the next slide. The polymers described above is the topic are formed by combination or linking of one or more type of monomer units. One or more types of monomer unit. So, in order to link these units together, various types of bonds are required depending on the nature and the type of macromolecules. Okay. First, look at the peptide bond. What is a peptide bond? A protein amino acids are linked by a peptide bond. Okay. Glycosidic bond. What is a glycosidic bond? It is formed between two carbon atoms of two adjacent monosaccharides. Two adjacent monosaccharides. A polysaccharide by linking many individual monosaccharide units. Many individual monosaccharide units. This bond is also formed by dehydration, removal of water. Okay. When you remove the water, it is called dehydration from the bond formation site. Okay. The next is phosphodiesterase. Phosphodiesterase bond. In a nucleic acid, a phosphate moiety links the third carbon of sugar of one nucleotide to 5 carbon of sugar of the succeeding nucleotide. Hydroxyl group of sugar is an ester compound. As there is one such ester bond on either side, so it is called as phosphodiester bond. Phosphodiester bond. Next slide. The chemical reaction together are called metabolism, removal of carbon dioxide from amino acid into an amine, removal of carbon dioxide from amino acid into an amine, removal of amino group in a nucleotide base, hydrolyze of the glycosidic bond in a disaccharides. Glycosidic bond in a disaccharide. Metabolic reactions take place in a series of linked reactions between as metabolic pathway. There is a series of linked reaction between them, so it is called as a metabolic pathway. The metabolic flow is called as dynamic state of the body constituents. Dynamic state of body constituents. Also, these metabolic reactions are always catalyzed reactions. They are catalyzed reactions. So, there is catalyst in it. The catalyst which hastens the rate of given metabolic conversion are also protein. So, the catalyst is also protein. These proteins with catalytic power are called enzymes. They are called as enzymes. These are divided into two main groups. Number one is anabolic pathway. These includes the formation of complex structure from simple ones. Example, formation of cholesterol from acetic acid. Formation of cholesterol from acetic acid. Okay. Protein synthesis. These are energy consuming pathway. So, they need ATPs, energy consuming pathway. Next is the catabolic pathway. Catabolism destruction. These include formation of simpler structure. The breakage of complex structure into simpler forms. Breakage into a simpler form. Example, conversion of glucose into lactic acid in skeletal muscle. Conversion of the glucose into lactic acid in skeletal muscles. These are energy releasing pathway. So, they release energy. Okay. Glycolysis. Glucose is degraded to lactic acid in human skeletal muscles. Lactic acid into human skeletal muscle, liberating energies. This metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called as glycolysis. 10, remember the number 10. 10 metabolic steps is called as glycolysis. This liberated energy is stored in the form of chemical bonds, and this bond energy can be utilized in various biosynthetic, osmotic and mechanical work when needed. Such a wonderful thing, isn't it? Let's move on to the next slide. Various chemical compounds are present at the concentration characteristics of each of them. These metabolites are in a steady state of metabolic fluxes. They are in a steady state of metabolic fluxes. Hence, the living system is kept in a non-equilibrium state by metabolic flux, which enables it to perform work as living organism. It has to work continuously and is unable to reach an equilibrium. It will not reach an equilibrium. The living state and the metabolism are synonymous and are correlated. They are synonymous and are correlated. Thus, metabolism and living state are incomplete without each other. You need metabolism and living state. Both are incomplete. Okay. The next example. The term enzymes are colloidal organic macromolecules which are mostly proteinaceous. Mostly they are proteinaceous. Enzymes are water soluble in nature, hence also known as biocatalyst. Biocatalyst. Each and every enzyme catalyzes the reaction of a specific substrate because of the configuration of the active site of that protein. Active site of that protein. Active site. It is a pocket of an enzyme into which a particular substrate fits. Pocketed enzyme where a particular substrate fits. Each enzyme functions at specific pH and specific temperature. Thermal stability. Enzymes thermal stability. It is an important quality of the enzyme. It is an important quality of the enzyme isolated from the organism who normally live under extremely high temperatures. Live under extremely high temperature. Example, hot thermal vents under seawater and sulfur springs. These enzymes have the ability to be stable and retain their catalytic power even at a higher temperature. That is the beauty of the enzyme. So enzymes are wonderful. Okay, let's move on. They enhance the rate of the chemical reaction without themselves being changed or used. Enzymes are efficient in very small amounts. They are efficient in very small amounts. Enzymes are highly specific as each of them catalyzes only a specific reaction. Example, maltase acts only on maltose. Enzymes can be denatured by heat. Human enzymes are active at 35 to 40 degrees centigrade. Denature at 50 to 55 degrees centigrade. These enzymes of bacteria living in hot springs have optimal temperature of 70 degrees centigrade. Okay. Next slide. Chemical compounds undergo two types of changes. The chemical compounds undergo two types of changes. One, physical change, or those in which, if you look at the physical change, what is the physical change? They are the in which the shape of the molecule or a substance changing without breaking of the bonds or change in state of the matter. Example, melting of ice in the water. Melting of ice in the water. So it's a physical change. So ice is melted into water. Both are same chemicals. Chemical changes are those changes in which bonds are broken and new bonds are formed during transformation. A general thumb rule states that the rate gets doubled or decreased by half for every 10 degrees. Every 10 degrees of centigrade changes either direction, either this side or that side. Catalyzed reaction take place at higher rate than that of the uncatalyzed ones. Next slide. It is the multiple step of chemical reaction in which each step is catalyzed by the same enzyme complex or different enzyme. Example, glucose to pyruvic acid. Okay. The same pathway also gives rise to a variety of different metabolic end products with one or two additional reactions. Example, under anaerobic conditions, listen carefully, under anaerobic conditions in our skeletal muscle, lactic acid is produced. Okay, lactic acid is produced. Whereas under normal aerobic conditions, pyruvic acid is produced. Aerobic is when there is a lot of oxygen in it. Pyruvic acid is produced. The same pathway during fermentation in yeast leads to the production of ethanol. Ethanol is ethyl alcohol. Okay. So move on to the next slide. During conversion of substrate into a product. Okay. During conversion of a substrate into a product, formation of an enzyme substrate complex takes place. Enzyme substrate complex takes place. The enzyme plus substrate is equal to ES complex. This complex formation is a transient phenomenon. A new structure of the substrate called transition state structure is formed. Transition state structure is formed. Stability is related to energy status of the molecule or the structure. Energy state of the molecule or the structure. In the graph, if you look at it, the y-axis represents the potential energy content and the x-axis represents the progression of the structural transformation. Progression of the structural transformation. When P is at a lower level than the S, reaction is exothermic or spontaneous. Okay. When P is at a higher level than the S, reaction is endothermic or energy requiring reaction It proves that whether it is an exothermic or an endothermic state, the S has to go through a much higher energy state. This difference between the average energy content of S from that of its transition state is called as activation energy. Okay. Moving on to the next slide. This hypothesis is also known as lock and key mechanism. It was given by Fischer in 1894. The catalytic cycle of enzyme action can be described in the following steps. First, the substrate binds to its active site of the enzyme. Fitting into the active site. The binding of the substrate induces the enzyme to alter its shape so that it can fit more tightly around the substrate. The active site of the enzyme now is close proximity of the substrate breaks the chemical bonds of the substrate. It breaks the chemical bonds of the substrate, due to which the new enzyme product complex is formed. A new enzyme product is formed. Next, the enzyme releases the product of reaction and the free enzyme is ready to bind to another molecule of the substrate. Ready to bind to another molecule of the substrate. Okay. Moving on to the next slide. Enzyme activity is altered or affected by the change in the conditions that can alter the tertiary structure of the protein. Tertiary structure of the protein. The factor includes, what are the factors that is going to include? It includes temperature, pH, change in substrate concentration, or binding of specific chemicals that can regulate its activities. Okay, such a wonderful thing, isn't it? Temperature and pH. Enzymes generally work at a narrow range of temperature and pH. Even with slight alteration, the enzymes will not work. Enzyme shows highest activity at a specific temperature and pH, known as optimum temperature and optimum pH. Example, amylase functions as 37 degrees centigrade. Okay. Concentration of the substrate. Concentration of the substrate. With the increase in the concentration of the substrate, the velocity of the enzymatic reaction rises at first the V maximum in the maximum velocity, which does not exceed by further rise in the concentration of the substrate. Next is the enzyme inhibition. Enzyme inhibition. The enzyme activity is also sensitive to the presence of specific chemicals shut off the enzymes. Shut off the enzymes. The moment they bind to the enzymes. Okay, they shut off the enzyme moment the enzyme binds to the enzymes. This process is known as inhibition, and the chemical responsible is known as inhibitor. The chemicals responsible is known as inhibitor. Example, magnesium 2+, calcium, hydrochloric acid, etc. Okay. These are the non-protein constituents of conjugated or compound enzymes. Conjugated or compound enzymes associated with the apoenzymes of the enzyme. Protein portion. Apoenzymes of the enzymes is the protein portion. As it is already studied that simple enzymes are composed of one or several polypeptide chain. However, there are a number of cases in which non-protein constituents, there are number of cases in which non-protein constituents called as cofactors bond to the enzyme in order to make it catalytically active. Catalytically active. Cofactor type. What are the cofactor type? Prosthetic group. Number 1 is the prosthetic group. Number 2 is the coenzyme group. Prosthetic group and the coenzyme group. That finishes biomolecules. It's quite tough. Probably I have to do it again. But it's a good chapter. You'll get full marks. All the best, kids. All the best.